You heard me say earlier that one of the characteristics of epithelial tissue is attachment, either to adjacent cells or to deeper tissues. These attachments are made possible by intercellular junctions, which are formed from interactions between plasma membrane proteins in two adjacent cells. Intercellular junctions are not exclusively found in epithelial tissue, but I'm going to be talking about them here because they are pretty critical for their normal function in most cases. And the first type of intercellular junction that I want to talk about is known as a tight junction. Tight junctions connect adjacent cells at multiple locations along their plasma membrane, and that holds them together very tightly, like their name suggests. In the picture that's on the left-hand side of this slide, which was taken under a microscope, you can see the tight junction protein Clodin labeled in green, and it shows up all along the shared border between the two neighboring cells. I always think of tight junction proteins as being like zippers that hold two pieces of fabric together very tightly. And that's because cells use tight junctions to form physical barriers between compartments. When cells are held together by tight junctions, it limits the movement of molecules through the spaces between the cells, so it limits paracellular transport. Another tight junction protein is known as Clodin. In the image in the lower right-hand corner here, Clodin is fluorescently labeled in epithelial tissue in the gut. And you can see in the top panel that Clodin is highly abundant by all of that fluorescence along the border between the cells. But in the bottom panel, they've given the gut epithelial cells a toxin isolated from Clostridium bacteria, which are bacteria that grow when food is kept at unsafe temperatures, and they're the most common cause of food poisoning. You can see that the presence of the Clostridium toxin causes the fluorescence to disappear in that bottom panel. And that tells us that the tight junction proteins are being broken down, and the barrier in the gut that would normally keep pathogens out is being weakened. So ultimately, this allows bacteria to enter your bloodstream through your gut and make you sick. So tight junctions are really, really important. Adherence junctions, on the other hand, join two cells together by connecting to proteins in their cytoskeleton, like, for example, the protein actin. So in this way, they're sort of like a drywall anchor that you might use to hang a picture or a bookshelf inside of your house. The connections are really reinforced. The last physical junction that I want to talk about between cells is a desmosome. Desmosomes join two cells together through proteins called connexons, and those proteins are associated with the plasma membrane. So if we think of tight junctions as being like zippers, then we can think of desmosomes as being like buttons, because they join cells together at focal points of stress. And they still allow for bending and twisting around those points of stress. So in contrast, if we had bending and twisting of cells that were joined together by tight junction proteins, we would most likely rupture the plasma membrane from all of that shear stress, because again, the plasma membrane doesn't have much structural integrity. So I mentioned that example because different types of epithelial cells might have only certain types of intercellular junction proteins, or in some cases, they'll have both. So it's specialization to serve their function. One desmosome-related disorder is known as Pemphigus vulgaris. This is an autoimmune disorder where the immune system actually attacks the connexons, the desmosome proteins. In the most severe cases, this causes blistering of the skin because the layers of tissue separate from one another, and then they become damaged by frictional movements caused by them rubbing against each other. The inflammatory response in the damaged tissue is really what creates the blistering. You can see that in the left picture, which is the histological image. You could check out all that white space there. And in the right picture, we can see how this manifests at the surface of the skin. Now, not all intercellular junctions are physical connections. Gap junctions are electrochemical connections between two adjacent cells, and that allows adjacent cells to share signaling molecules and ions. So in essence, they provide a way for adjacent cells to sense what's going on with their neighbors, in the same way that you might be able to sense what was going on with your neighbors if you share a common entryway or a wall. We're actually going to talk a lot more about those later when we get to our nervous system unit and when we get to our muscle unit. Epithelia can also be classified anatomically, but fortunately, this is a much more straightforward classification scheme. To classify epithelia in this way, we really have to think only about the shape of the cell and the number of tissue layers that are present. So simple epithelial tissues contain only a single cell layer, whereas a stratified epithelial tissue contains multiple cell layers. Thinking more about shape, squamous epithelium are thin and flat, whereas cuboidal epithelium are cube-shaped. Columnar epithelium take on a rectangular appearance. Now, there's also a few special cases. So, for example, transitional epithelium often appear as both cuboidal and columnar, depending on how stretched or how distended the tissue is. In other words, if you stretch a cube, you can make a rectangle. Pseudostratified columnar epithelium appear as a multi-layered tissue, but in fact, they're actually a single layer of tissue. The giveaway to identify pseudostratified tissues is to look at the location of the nuclei. In a true simple columnar tissue, the nuclei and adjacent cells often line up uniformly in the z-axis, whereas in a pseudostratified columnar tissue, they tend to be all over the place and a little bit more staggered. To give you one example of how the anatomical and functional classification schemes overlap, in many cases, pseudostratified epithelium are also ciliated. So in cystic fibrosis, which we talked about already when we were together in class, we know that cystic fibrosis typically occurs because of mutations in chloride channels. Those chloride channels are found in these cells. And the solute movement across the plasma membrane is impacted. That leads to a thickening of the extracellular fluid because of osmosis and eventually to mucus buildup. But this actually impedes the movement of the teeny tiny cilia, and that impacts the function of tissues that have pseudostratified columnar epithelium, like the respiratory system and the reproductive system. And so, left untreated, this can often lead to severe respiratory distress due to the buildup of debris in the airways, and it can also lead to things like infertility because of the impaired movement of sperm through the reproductive system.